Mitosis by Kinsley and Rachel. Interphase. Interphase is the interval between the end of one mitotic or meiotic division and the beginning of another. The cell spends most of its time in this phase. During its time here, it grows, replicates its chromosomes, and prepares for cell division. Gap 1. The first of 4 phases. The cell grows and synthesizes mRNA and protein. S phase. The cell synthesizes a complete copy of DNA to the nucleus. It duplicates centrosomes, which separates DNA during M phase. G2. This is the shortest phase during which the cell continues to grow and produce missing molecules it needs to divide. It also duplicates its organelles to each daughter cell so it has what's needed. When/how mutations occur. When a mutation occurs. If the mutation is a gene mutation, it occurs in the S phase. If the mutation is a chromosomal mutation, it occurs during metaphase or anaphase. How a mutation occurs. Gene mutation. Mutations occur when there is DNA copying mistakes made during cell division, exposure to ionizing radiation, or exposure to mutagens. Chromosomal mutations. These mutations mainly are due to abnormalities in the chromosome structure.
Anaphase. Anaphase is the third stage of mitosis. The sister chromatid of the chromosome has spindle fibers attached to it. The spindle fibers shorten. As a result, the sister chromatids are separated from each other. This process is called disjunction. As the spindle fibers shorten and pull to the opposite ends of the cell takes on a V shape. Telophase. Telophase is the last stage of mitosis. It separates the duplicated genetic material from the nucleus of the parent cell into the two daughter cells. During this phase, 2 identical nuclear membranes are formed, separating the 2 new cells. The chromosome decondenses and the kinetochore disassembles, as its job is now done. Cytokinesis. While cytokinesis isn't a part of mitosis, it's part of cell division. The cytoplasm of the parent cell is divided in order to separate the new cells fully. A loop of protein filaments, called a contractile ring, is placed around the equator just under the plasma membrane of the separated cells. The loop of filaments shrinks around the equator, pinching until there is 2 cells separated by their own plasma.
How the cells could become cancerous. Cancer cells generally form after mutations of the cells bypass the cell checkpoints. These cancer cells grow unchecked and uncontrollably. Once one of these cancer cells have bypassed a checkpoint with its mutation, it is cancerous and will continue to divide and grow, which could cause severe damage to the body. How scientists are fighting cancer. Although there has yet to be a cure for cancer, there has been many studies and many treatments created over the year. Some cancers, if caught at an early enough stage, can be surgically removed by cutting out the cells so they can't divide or infect the body any longer. Most of the time, cancer cells are treated with chemotherapy, a treatment which utilizes a variety of anti-cancer drugs. Hormone therapy can be used to eliminate or reduce the amount of hormones being produced in the body to slow the spread of the cells. Radiation therapy or radiotherapy uses radiation to attempt to shrink or eliminate the cancer cells completely. Immunotherapy is the process of strengthening the body's natural immune system and its ability to fight the disease on its own. Immunotherapy has shown consistent success in treating 15 different types of cancer. Target drug therapy is the process in which doctors take a certain cancer-killing drug and inject them into the infected areas. This, used with a combination of other cancer therapies, helps doctors control the growth, cell division, and spread of cancer cells in the body.